So we're in the middle of looking at genetics, and when we think about genetics and we think about heredity, we think back to our parents and how am I like my parents? Am I tall? Am I short? Do I have the same eye color? Do I have the same skin color? And so one of the things that we have to remember is that genetics can be really complex. As you're learning by looking at the eye color study, most human genes, eye color, skin color, hair color, height, weight, all of those things are usually determined by more than one gene. What Mendel did, remember, to be successful was look at an organism where the traits, like flower color, were determined by one gene, and where that gene came in just two forms, the forms that we call dominant and recessive. And by examining this model organism, the pea, Mendel was able to come up with these three main principles. The principle of dominance, the principle of segregation, and the principle of independent assortment. Now these principles hold true in most cases. We will be examining in this unit some cases where these don't hold true. But for the most part, you really need to know these three principles because they explain how organisms inherit traits, at least the traits that are genetic. Remember, if you think of something like the language you speak, that's not genetic. When we look at Mendel's theories, one of the important things to do is to be able to apply Mendel's theories to make predictions. And so it turns out that Mendel did his work in the mid-1800s. Right around 1860 was when Gregor Mendel was completing his research. However, his research wasn't really commonly well known or widespread. It wasn't until a man named Reginald Punnett in about the 1900s stumbled upon one of Mendel's books, and Punnett was actually a mathematician as well. So he was a biologist, he had a degree in zoology, but he was also really interested in math. He was a mathematician. And Punnett is actually the one who created a system for understanding Mendel's principles and how these principles were applied. Remember, Mendel has three principles, so we're going to look at those one by one. So what I'm actually going to do is I'm actually going to cover up part of this picture, because if you try to look at the whole picture, it's pretty confusing. But if we start by just looking at the top of this picture, if we just look here at the P generation, the parental generation, we can actually see that the parent will produce gametes. Now remember, this happens during meiosis, but this production of gametes is actually the principle of segregation. And this principle of segregation is what explains that each parent donates half. And so what Mendel's principle of segregation explains is that even though a parent has a genotype like big R, or homozygous dominant, that the gamete will only have one of those. And so when we look at this principle of segregation, the Punnett Square is actually going to, if you scroll down again, help us remember how each parent only gives half. So we're going to draw the Punnett Square just for the R's. If we look just at the R's and we see a parent that is big R, and another parent that is homozygous recessive. So we're going to start trying to use our correct vocabulary. Before we might have said big R, now we'll call this homozygous dominant. So Reginald Punnett was looking at Mendel's principles and looked at how each parent really only donates half. And so he set up this chart to represent the probability of this parent donating this R, or the parent could donate this other R. Then we have these lowercase r's. And so the key to the Punnett Square is that each parent is still only donating half of their original genotype. And that's the principle of segregation, separating the homologous pairs so that each parent donates half. Now fertilization comes into play, and this is where we see the possibilities of the new offspring. It turns out for this cross and this mating, all of the offspring would be exactly the same. All of them would be heterozygotes. But the interesting thing is, well, they have a recessive gene and a dominant gene. And so this is where Mendel's principle of dominance comes into play. If we look at Mendel's principle of dominance, what we see is that if you have the dominant allele, just like here, if you have the dominant allele, you will show the dominant phenotype. And that is Mendel's principle of dominance. And so when we use it in a Punnett square, we're actually able to see segregation, the separation of the homologous pairs. We're actually able to see the principle of dominance. And what we're going to see in just a moment when we look at the dihybrid is the principle of independent assortment. But it is really important that you understand that this Punnett square really helps us apply Mendel's principle. The principle of segregation, each parent donates half, and then the principle of dominance. And so Mendel's principle of independent assortment, remember, it tells us that genes on separate chromosomes are inherited independently. And so what this diagram shows you is a hypothesis of dependent assortment, which is not true, and a hypothesis of independent assortment, which is true. So when you're examining this diagram, what's important here is that we're actually looking at two traits. So independent assortment means the assortment of more than one trait. In this instance, we're looking at both P color and P shape. This parent was yellow and round. This parent was green and wrinkled. And so when we crossed these two parents, it didn't matter. All of the offspring got a dominant Y, and so they were yellow. All of the offspring got the dominant R, and so they were round. And so we have this hybrid here, which is hiding these traits. However, if we cross a big R, little r, a big Y, little y, times a big Y, little y, and a big R, little r, times a big R, little r, we let them self-fertilize and mate, we do not get this ratio. We actually, because of independent assortment, will see that we get a much wider variety. Because there's a chance that this big R will end up with this, this big Y will end up with this dominant allele, and then we would get big Y, big Y. However, this trait of color is separate from the trait of shape. So I could be yellow and round, or I could be yellow and wrinkled. I've never had a yellow and wrinkled parent. But because of the way that these assort independently, the offspring could inherit any of the combinations of these alleles. And so we're going to be calling this a dihybrid. And we'll be practicing dihybrids and looking at even more traits. But it's Mendel's principles that are applied when we look at Punnett squares. And so one of the things that you really want to hold on to is, what are Mendel's principles and how do we use a Punnett square? It's pretty important to know that the Punnett square represents meiosis, the separation of the pairs, fertilization, the chance that this gamete is fertilized by this gamete, and the ability to then make predictions after we have our Punnett square results. So the final part of these notes is really looking at what predictions can you make. Before you move on, you do just want to take some time and go back and practice what is segregation, what is dominance, and what is independent assortment, and how is it shown here. The final part of the notes reminds us that Mendel's principles allow us to make predictions. When we make predictions, we do have to remember that we're talking about probability. And so when we examine probability, we remember that there is no certain outcome, but that we have chances that certain things will happen or occur. So think back to flipping the coin, and if you flip a coin 10 times, you might get 5 times heads, you might get 6 times heads. So one of the things that we are going to look at is, how do we make a prediction, and how do we compare the actual outcome, the observed, to what we expected? So you'll see chi-square a lot during this section. One of the things that we want to be really careful with, though, are these words phenotype and genotype. Remember, phenotype is the physical appearance, and this physical appearance depends on the genotype, which is the genetic representation. And all of this is because of Mendel's principles and the Punnett square. So what we see here, this picture, is actually reminding you the differences between genotype and phenotype. You should be able to work backwards and actually predict what the parents were if these are the ratio of the offspring. And so I just want to take a moment and help you remember how to work backwards in these problems. If I have some offspring that are purple and some offspring that are white, the only way to get white is to be homozygous recessive, which means that this allele, if I want to have an offspring that is white, I had to get a little p from one parent and a little p from another parent. So this means that both parents have to have the recessive allele. Now here's a question. Are both of my parents heterozygotes? Are both of my parents... So actually we could kind of write all of this out. We could have big P little p times big P little p. That would give us white. We could have little p times little p little p. That would give us white, but only all white. So this means that this could not be the parent. Another way would be to be big P times little p. That would give us white, but if we create the ratios of this, if I look at the ratio of 3 to 1 and 1 to 2 to 1, this Punnett square, if you work this one out, big P times little p, this actually gives us a ratio of 1 to 1. This is the ratio where you will get a 3 to 1 ratio. Big P, little p, big P, little p. I struggle with the p's. And this is the only one that would be white. And so we need to be able to make predictions going forward and going backwards.